Well, it's my pleasure today to introduce today's speaker, Jocelyn Wardrup. Jocelyn is a proud native of the state of Delaware, where she studied to get her bachelor's degree. She actually has a degree in geography and in soil science. After she did that, she went to work, joined the environmental consulting force, and for about 7 years worked doing soils and wetlands work. After that, she came back and did a master's degree at the University of Delaware looking at carbon storage in Atlantic coast marshes. And she finished that up in 2021 and then started here on a doctoral degree. She's working on manganese oxide. She's going to tell you a little bit about that. She's in sort of the final stages. She's in the process of writing her dissertation and hopes to graduate towards the end of the summer. She also does carry credentials as a certified professional soil scientist and as a professional wetland scientist. And while she was here, she's received a number of recognitions, but one that's especially important, especially in light of the announcement that we heard. I mean, 3rd place at the National Soil Judging Contest is really outstanding. Yes, it is. I always promote that to the dean as like a Final Four appearance. Yeah. Right? So, but two years before this, when in 2023, Dr. Needleman brought Jocelyn on board, not as an assistant soil judging coach, but as a co-head coach to the soil judging team. And they took the team out to Oklahoma, where they won the national championship. And so there are very few students that have that on their record. So that's certainly an important recognition. But today she's going to talk to you about what she's been doing in her research the last few years. So I'm sure you'll all enjoy it. Yeah. Jocelyn?
Thank you. Greetings, thank you all for being here today. I'm going to present to you some of my dissertation work, the distribution, formation, and mineralogy of manganese redox features in soils. So before we get into manganese in soils, I thought I would enlighten you with some fun facts and background on manganese in general. Manganese is made in the universe in stars, dwarf stars, supernova stars, and there's some of it present in cosmic rays. On Earth, it's the 10th or 12th most abundant element, depending upon what source you look at. Crustal rocks average 0.1% of manganese content. It's an essential element for both plants and humans. I think essentially all microorganisms. In humans, the average human body contains 12 milligrams of manganese. It's concentrated in the skeleton, your kidneys, liver, and then it is an important cofactor for enzymes, and it plays a role in blood clotting. So it can save your life. Lastly, manganese pigments, they're dark black in color. They've been found in some very ancient cave paintings. Perhaps you've seen this very famous one that's estimated to be 17,000 to 20,000 years old in France. Manganese is a transition metal, atomic number 25 with the average atomic mass of 54. It's positioned between chromium and iron, and you'll see here soon that there are some neighboring interactions with these elements. In natural systems, manganese has the oxidation states of +2, +3, and +4. It also has the oxidation states possible of +2 to +7 in some non-natural systems. And that's more than any other transition metal. And because of this, it has the ability to gain or lose electrons while maintaining a solid phase. It's characterized as having small crystals with large reactive surfaces. However, it's poorly crystalline, and they're very small nanoparticles of around 5 to 100 nanometers in size. It's involved in a number of favorable reactions. It can absorb lead, cadmium, copper, zinc, arsenic, but then it can also have some unfavorable reactions, such as the oxidation of chromium from chromium-3 to chromium-6, which is a carcinogen in groundwater and the subject of the environmental law movie Erin Brockovich, which is a really great film. For this talk, I wanted to share with you that I'm going to be using the term manganese oxides. However, I wanted to define that to share with you that I'm using this term collectively as manganese oxide, hydroxide, and oxyhydroxide. And this just simply is manganese bonded with either oxygen, hydroxyl, or an oxygen and hydroxyl ions. So now that you know some of the basic chemical properties of manganese, let's dive into manganese properties in soils. On the left here, you'll see the general reaction of manganese in soils. Manganese in the oxidized form on the left, a +4 form, and then manganese in the right, +2, and that is a colorless soluble form. The process of reduction and oxidation in soils can generally be described by the redox ladder. The redox ladder shows that when oxygen is removed in the soil environment, the pedosphere, assuming the presence of carbon, microorganisms available to respire, that the microorganisms need alternate electrons in the absence of oxygen. So they reduce in sequence nitrogen, manganese, sulfur, and then carbon. This process is influenced by EH and pH conditions within the soil. And this diagram specifically shows when these chemical reactions take place. The main takeaway from this slide is that manganese is characteristic of moderately reducing conditions, whereas iron, sulfur, and carbon are characteristic of more strongly reducing conditions. And so for manganese to be reduced and concentrated in soils, and for the redoxomorphic features I'm interested in my dissertation to form, these chemical processes have to take place, at least oxygen, then nitrogen, and then manganese needs to be reduced and then reoxidized. So now that you understand how they form, here are the different types of manganese redoxomorphic features I have identified to be present in soils. They are coatings, concretions, nodules, and masses. The black and white diagram shows an idealized landscape setting of where the soils occur, or where I would expect these features to occur. The mottled zone is where iron redox features occur within the soil, or the general long-standing seasonal high water table. Coatings we would expect to find in more well-drained systems because they form from brief periods of wetness and then oxidizing on the outside of head faces, whereas concretions, nodules, and masses have been documented to form in more moderately well to somewhat poorly drained systems because they need either perching conditions or manganese being enriched within the groundwater to create those features. Please note that manganese features form usually above iron features within a soil profile, and so you may have manganese features and iron features within a profile. However, they can form in different zones, and they're not synonymous with each other. To give you some characteristics of these types of features, coatings are thin film-like coats that form on the outsides of soil beds. Concretions, nodules are sand to gravel size, circular or acircular pellets within the soil, where if they have banding within the interiors, we term them concretions, or if they are one solid color on the interior, we define them as nodules. Masses are soft, non-coherent, grainy accumulations of manganese that occur within soils. So the overarching goal of my dissertation is to just understand more about these features that have not been well documented and advance knowledge specifically on 4 different categories. The distribution, where do these features occur since they do not occur across all soils? How do they form is the literature that is available supporting what we find on how they form in soils. What is the mineralogical makeup of manganese in soils? And then how can we help soil scientists in the future with the identification and description of these features? Is what's available accurate, and should we still be using that criteria, or should we have some fine finessing to that? I have 5 chapters as part of my dissertation. Today I'm going to go over 2 of them. And diving right in, here is chapter 2. Chapter 2 is a synthesis of the available information for manganese redox features in soils. This was my database mining and mapping chapter. For this work, we used the USDA, NRCS, the Natural Resources Conservation Service, and the National Cooperative Soil Survey data set to look at the available soil maps, and then we used R, RStudio, and Excel to process the data and make resulting soil maps. The research questions for this work are: what are the broad regional spatial distributions of various manganese redox features in soils that can be extracted from the public database? And then, secondly, does this information shed light on pedological processes involved in their presence or formation? So some of you are aware the soil survey in the US has been going on for 125 years. As a result, the data collected has been being updated and getting better and better with time. There's a robust data set to work with and to understand the soils across the US. This data set includes soil maps. Soil maps contain soil series, and there are over 24,000 soil series now across the US. I'll go over what a soil series is next, and then there's also extensive soil lab data for about 20,000 pedons. Soil series are the most specific level in soil taxonomy. Here's an example for the monalto soil that you'll see later on in this presentation. There are 12 general soil orders going from general categories in soil taxonomy down to the most specific level, which is the soil series, because it contains so much additional information. This is an example of an official soil series description. The term series and OSD, official soil series description, I'll use interchangeably in this talk. They essentially mean the same thing. And this is an example of some of the different pedological and environmental information that you can gain from looking at an OSD. Here's an example of the extent of that same series, monalto. For all of these active soil series, there are spatial and extents and geographic data related to those soil series. Here's an example of a soil map. Perhaps some of you know the Central Maryland Research Farm nearby Glen Elg, Maryland. Maps are made up of polygons, and they have units within the map. Here's an example of a unit within the map, and they're named for one or more soil series. This one is named for Glen Elg. And here I show you that despite it being named for one soil series, there are multiple soil series within the mapping unit, and these are termed components because there is a possibility that these soils may occur within that mapping unit, and these components are important to remember when I present the results of this research, that there's different percentages of possible soils within the mapping unit. So the justification for this work is that since existing studies do not provide information on manganese redox features, and there's this long-standing 125-year-old great data set to work with, we decided to use the OSD data to mine the soil series information and map soils with manganese redox features across the US. I wanted to provide some previous studies that were available before diving into the results to let you know what was available. Here's manganese concentrations in well water data. The map on the left is the McMahon et al. Map, and you can see that there is some good information there. However, it's a little difficult to make some assumptions and conclusions about some trends across the US. On the right is the Giuseppe et al. Map, which shows manganese concentrations with groundwater in North Carolina, and you can see that there are some definitive trends relating to manganese concentrations in groundwater and parent material / physiographic region. Here's a map of manganese subsurface concentrations that was produced from Smith et al. From the USGS. This map shows manganese contents and not manganese features. I've created a simple legend here to show you that the areas in blue are less than 1/1000%, and that the areas in red are greater than 0.1%, which I want to stress that 0.1% is still very low manganese content in soil. What you see here is a good, reasonable picture of what you would expect to find for the manganese contents in the subsoil, and it may be helpful for understanding if manganese is tied to parent material. Lastly, here's a map from Jiang et al. That looked at iron manganese features in loess soils of the Midwest. While this was an interesting geographic resource, they did not differentiate between iron and manganese features, and so we were not sure what features on this map were definitively manganese and not possibly just iron. And this map only focused on one parent material in one region of the US, where we wanted to look at across different parent materials and also see how things were in our area, the Mid-Atlantic, where we're more familiar with the soils. Therefore, the knowledge gap, as I've shown you, is that it entails answering how extensive these manganese redox features are across the US. To walk you through the methods that I used to achieve the results, I searched the terms manganese, iron manganese, and mangan, and then spelled out manganese through the OSD database using an R code to capture the descriptions that likely had manganese redox features. And this resulted in about 10% of the available soil series, so 2,400. Next, I used the code to extract all relevant soils information and create a table of attributes, a data table of sorts. And so while this isn't a complete list, I was able to obtain some soil classification information on parent material and drainage class to name a few soil characteristics that may be of interest for understanding where manganese redox features occur and why. One limitation with the database is that it was so large that that soil series, you cannot mine the soil series and just tell the code to just find the line where manganese redox features are described and tell me what horizon, what depths, and what texture is documented there. That's a very hard thing to tell the computer processing code to do. So we determined that these series where we wanted to obtain that information and what types of features needed to be manually reviewed. However, 2,400 sounded like a lot of soil series to review manually. So we trimmed that to be soil series greater than 10,000 acres, removing 1,000 soil series to something slightly more manageable of 1,400 to go through. Then I manually went through these soil series and searched the terms and then extracted some relevant pedological information such as the horizons, the depth, and properties relevant to the features such as their type, how many were there, their size, their color documented, and then save that into a data table to then look at. I then created maps of the results to show the spatial extents of the entire data set, the trim data set, and then the individual types of manganese features. And then not only did we have this spatial data set, but we had this data table where we could look at the proportions of soils and the manganese redox feature types and look at them against each other and see if there were any trends that stood out, such as soil taxonomy, drainage class, soil temperature regime, to name a few. With the help of our esteemed collaborator at the USDA NRCS, Dr. Dylan Bodette, we've compared some of the results to the NASA soil database, which is the National Soil Information System. This is an internal USDA database that's the most extensive soil resource available to use. We've completed part of this work, however, the analysis is still in progress. Compare the results to other spatial data in previous studies. And then lastly, provide guidance on redoxomorphic feature identification and description for soil practitioners moving forward. This step is to be completed. However, we have some thoughts. So here are my overall findings. So recall back to the introduction where I briefly introduced you to the concept of percentages and components mapped in soil mapping units. Well, these maps show the percentage of components mapped with manganese features described in the OSD. The map on the left are all of the series descriptions with manganese in the OSDs, and then on the right you have my trim data set where I looked at just soils that were greater than 10,000 acres. And you can see that these maps are very similar, which I found very reassuring that by trimming the data set of over 1,000 soil series, I was still able to maintain and capture the general picture and theme of what the data was trying to tell us. The white areas of these maps show you where no manganese features are mapped. The blue areas show you where up to 15% manganese, 15% of the components of soil series may have manganese described within the OSDs. And then the teal and turquoise areas show 30 to 50% where series may be mapped as components with manganese redox features. And then there are a few isolated red pockets where it's 80 to 100% of the components mapped that had manganese described within the OSD. And this is along the Gulf of Mexico coast, between Houston and Corpus Christi, up nearby Toledo, Ohio, and then nearby Champaign, Illinois. So what can we make of these maps? Of course, I have a few thoughts to share with you today. The map on the left corresponds to climate, showing the ratio of evapotranspiration and precipitation, and then the map on the right is showing soil moisture regimes across the lower 48, the conterminous US. Should I just, maybe it will go away. For a resulting map of manganese redoxamorphic features, it seems that the western boundary is corresponding to this Udic-Ustic soil moisture regime line that we've drawn out on this map, and then we've also replicated on the two other maps here. Soil moisture regime is related to climate. The two maps up there are communicating some very similar things related to climate. The geographic boundary supports what we think about manganese redoxamorphic features, that they need the moisture to become reduced enough in the soil to then become reoxidized. So on the right-hand side of this line, you have soils that are humid to semi-arid, and then on the left-hand side of that line, you have arid soils where there's not enough moisture for the manganese to reduce and then reconcentrate. And so that may be the main description as to why there's not that many manganese redox features described in OSDs to the left of that line. So what about the eastern boundary? The eastern boundary I found to be related to sand content. My apologies for these grainy maps. I'm working on finding a better resource, but I thought that these were good for the purposes of today's talk. They're generated from USDA data, and the sand content describes well, especially the southeast, and the soil texture content in the upper left there describes well for the gaps in New England with why there may not be manganese redoxamorphic features there, because there's just too great a sand content possible, and that there may not be the evaporative mechanisms to form the manganese redox features there. I wanted to show you that comparing these results to the manganese concentrations in the sea horizons of the USGS map, that it corresponds well to the gaps in manganese redox features along the southeast here. However, in the upper northwest, there are some gaps there. And so the take-home message from this map is that some things can be informative for describing why manganese redox features are described in one place and not another. However, it appears that there needs to be multiple factors in the creation of features. And so just because there's a higher content of manganese in the northwest of the conterminous US does not mean that manganese redox features are going to form there, because it does not have the suitable climate and the moisture for those features to form. So it appears to be a multi-variable dependent process. Here are the findings for the different types of manganese redox features: the coatings, concretions, nodules, masses, and then the combination category is more than one different type of feature. While I can't say much about the concretions, nodules, and masses for their extents, I can say that the coatings, the extent of the coatings seem to be more selective and limited, and that for the percent of the components mapped, that there seem to be far fewer OSDs that have the manganese features mapped in them versus these other two feature types of concretions, nodules, and masses. While we would like to think of the description of manganese redox features to be uniform across all of the US, and that soil scientists may be trained similarly, there are some artifacts in the data that you can see here, and this may reflect when soil surveys were done at the state and county level. Now they're done in physiographic regions where they're not at state-county boundary lines. These artifacts, fortunately, though, do not show up in the main picture of the results, and they're just an artifact that show in these results when you extract the different types of redoxomorphic features. Lastly, as a note about the terminology, some of the terminology in the OSDs was not as precise as we would have wished. A common term that was used was accumulations, and so I equated that to the masses category, and you just have to use a strict approach and just say this is going to be this and this is how I'm moving forward, and not, you know, interpret each one separately. So I really hope that a person describing things as accumulations really meant for them to be masses and not coatings, but you just have to take it and move forward. I wanted to tell you a few results of the tabular data. So we had the spatial data, and then we also had the tabular data, and that reviewing the tabular data, I can synthesize that manganese redox features are most commonly formed in humid climates, fine textured soils, parent materials derived from sedimentary rocks such as limestone, loess or alluvial deposited soils, soils with slightly acid or neutral pH, BT horizons, zones of clay accumulation and active pedogenesis, and then there are typically only less than 2% of these manganese features described within a horizon. In reviewing the different types of manganese redoxomorphic features in the tabular data, I really didn't find that many differences across the different feature types. However, one difference I found was that, similar to the diagram I showed you where we thought that coatings would occur in more well-drained soils and that the other features would occur in more poorly, somewhat poorly drained or mildly well-drained soils, the data supports this. And so coatings were proportionally in moderately well and well-drained soils, whereas the other types were spread out among poorly to well-drained soils. Note that hardly any manganese redoxomorphic features were described in excessively drained soils. These are soils that are commonly, you know, like think of a pile of sand. You put water on a pile of sand, it's going to go right through. There's no process for that manganese to become reduced and then oxidized. And then in very poorly drained soils, you can think of just water being stagnated, and the manganese is just in soluble form and not able to form because it's just so wet. So some preliminary conclusions from this chapter are that we provided the geographic extents of manganese redoxomorphic features across the conterminous US, how these redoxomorphic features are commonly described, provide some pedological information for the different types of manganese redoxomorphic features, and we've identified some soil properties where they may be likely to contain or potentially maybe develop manganese redoxomorphic features in the future if they're young soils. With that, I'd like to dive into the second chapter I'm going to present today, which is, can these features be developed in a lab? So here are the different feature types that I'd shown you before. Concretions nodules have been a subject of fascination with the soil chemists. However, little efforts have been done on manganese coatings. There's some literature on the coatings, and what the literature tells us about what the coatings need to form is that there needs to be some manganese present within the soil, and they need to have brief periods of wetness followed by longer dry cycles. This process was detailed by Professor Emeritus Fanning and his late wife Fanning in their book Fanning and Fanning with their type 1 model. where they describe the reduction in the interior and the moisture potential gradient causing the water movement, and that this causes manganese in +2 form to move with the water and then oxidize at the soil surface, creating that manganese oxide coating, that striking black color of that redox morphic feature. The research questions for this work are, can evaporative drivers facilitate soluble manganese being concentrated at the PED surface where it oxidizes and can form a manganese coating? And then secondly, are there soil properties that may facilitate or inhibit the formation of manganese coatings in soils? So to walk you through the methods of this part of the experiment, we decided to use 6 soils of the Mid-Atlantic region that I've highlighted here. Soils with asterisks, the Mont Alto, Hagerstown, and Glenelg, all have manganese redox morphic features described in the OSD, and we have also observed them on our site investigations. These soils represent a variety of physiographic regions and different parent materials in the Mid-Atlantic. Mont Alto, Glenelg, and Codorus are Piedmont soils. Hagerstown is a soil developed in residual limestone rock. Downer and Elkton are coastal plain soils. Downer is well drained, Elkton is poorly drained. We constructed these imitation soil PEDs, which are approximately 3 by 3 centimeters in size, attached geotextile from the bottom. And the goal with this was that we would encourage saturation to happen from below to mimic groundwater table systems within soils, and then encourage the evaporative processes to happen at the surface where we can observe them and document them. By using these small little imitation soil PEDs or ISPs, we were able to test more soils at once and have a more streamlined experiment and not something that was very bulky. So next, we saturated these imitation soil PEDs with laboratory solutions for 24 hours. We used manganese nitrate, manganese chloride, manganese sulfate, and potassium permanganate. We also had a control with just water. Manganese nitrate, manganese chloride, and manganese sulfate all have manganese in +2 form, where in order to form the coatings, the manganese has to oxidize. However, potassium permanganate, manganese is in +7 form, so it actually has to reduce to +4 to form the manganese coating. So it's a little bit of a reverse. You don't expect to find potassium permanganate in nature. However, with these, the idea behind this was use available lab solutions and see what happens. So these are the 4 commonly available lab solutions where manganese could be in a soluble form to test. After saturating the solutions for 24 hours, this is the general process of the experiment. So you have a wetting cycle used with deionized water, and then a drying cycle where they air dry at room temperature for 6 to 7 days, and then it repeats. And we weigh the imitation soil PEDs to obtain the % water in between to get an idea of how wet or how dry they are and have some documentation of that. There were 15 wetting and drying cycles completed, and you can see that the soil moisture behaved rather similarly across all soils tested. Changes in surface color were documented using a colorimeter that documents Munsell soil color to the nearest tenth of a value for hue, value, and chroma. For those of you not aware, soil scientists use the Munsell system to document soil color in soils, and it can also be quantitatively done with this colorimeter. We then impregnated selected imitation soil PEDs where the surfaces of the imitation soil PEDs had strongly expressed manganese coatings that formed. We sent them off for billets to make thin sections, so that way we could look at them under the microscope and look at the thickness and any other properties we were interested in. Lastly, we collaborated with a partner at the Smithsonian, Dr. Jeffrey Post, who is a wonderful expert in manganese oxides, to use their Raman spectrometer to evaluate the manganese oxide contents within the samples of the strongly expressed surfaces. This instrument is housed at the Mineralogical Sciences Department at the Smithsonian Museum of Natural History in DC. It uses a microscope to achieve nanometer scale and 4 different lasers, as Dr. Post terms it, to interrogate the samples. It gives a mineralogical fingerprint of the mineralogical makeup of the sample, and Raman spectroscopy is uniquely suited for manganese oxides because the laser beam is being absorbed by the entire black color of the sample, and so you're just getting back exactly what you want, especially since coatings tend to be very thin, and you're not getting any noise in the data from underlying clays below. Here are the results of this experiment. The visual results here, I've highlighted where the manganese coatings were formed most strongly. They were formed in soils that happen to already have manganese features and in the solutions of manganese nitrate and manganese chloride. You can see below the control did not form any coatings. Potassium permanganate did not form any black coatings. Manganese sulfate resulted in the accumulation of sulfate salts, which were primarily gypsum. And then for the manganese chloride and manganese nitrate results, the top 3 right samples of Monalto, Hagerstown, and Glenelg resulted in the strongest coatings, and that the Cadorus resulted in weak coatings, and there were no coatings in Elkton or Downer, despite them being formed in the other soils of that same treatment. I provided you some close-ups of the Monalto soil treatment at the 7 wet-dry cycles. The 3 columns there show the wet-dry cycles at 5, 10, and then 15, and you can see that they get progressively darker with time for those results where they were exceptionally dark and well expressed. To show you the results of color changes, here's a graph of the starting and ending colors. You can see the bottom 3 lines representing Monalto, Hagerstown, and Glenelg, and that they had some very drastic color changes. And then to put this on a Munsell book, here were where the starting colors were, and the ending colors were actually a 0 chroma. They weren't even a 1, so that's why it's kind of pushed over. So it was even darker than what that page shows. Here are some pictures of the micromorphology analysis. We primarily use thin sections to determine coating thickness, and we determine them to be 25 to 55 microns thick. And you can see the coatings that were formed on the surfaces of the 3 strongly expressed samples. Here are the mineralogical results for the soil treatments that formed the strongly expressed coatings. Through Raman spectroscopy, we were able to determine that they were primarily poorly crystalline layer structure phases of lithiophorite, bernacite-like oxides with some tunnel structures present. For the soils where we had good coatings form, the pH of those soils, whoops, I thought I had those boxes highlighted, I'm sorry. The pHs of those soils tended to be above 5.5. And then the organic carbon content tended to be low, less than 0.4%. The extractable iron content was greater than 2.8%, and there appeared to be some need for manganese content of 0.008% or higher. And for all 3 samples where the manganese coatings formed well, the Monalto, Hagerstown, and Glenelg, they had higher clay contents of greater than 34%. However, you can see 2 other soils here had particularly high clay contents and then a higher manganese content, but they didn't form strongly expressed coatings like the other 3. And so we think that it's a multivariable process for there to be multiple soil properties needed to form manganese coatings, and not just one property present for a soil to develop strongly expressed manganese coatings. So to provide some conclusions of this work, by creating manganese redoxamorphic features in the lab, we were able to demonstrate the importance of moist conditions followed by drying cycles. This wicking process, we really demonstrated that well of what the literature documented, and then showing that in a laboratory setting. and that inherent soil properties such as what we just went over, the higher pH, the lower carbon content, higher iron, higher manganese content, and then higher clay content, that may be a recipe of success for creating manganese redoxomorphic features in soils, and that combination may be needed, or maybe parts of it. So looking forward, maybe another student will look at different combinations of some similar soils with these properties and determine if all those are needed or if only a few select ones. So with that, I'd like to provide some acknowledgements for the many people that contributed to this work. Without them, this work would not have been possible. There were many sites and a lot of great collaboration with the NRCS. Here you can see the state soil scientist of Virginia hunting for concretions for me, Dr. Wilmot in the pit too. So it was great. It was a really fun time. This gentleman used to be an assistant coach for soil judging here. I guess he got his graduate degree, Carl Robinette here. So that was awesome. Thank you especially to my advisor. It's really been a wonderful experience to study under Dr. Ravenhorst. It was a dream for a long time, and it's been amazing. I just wanted to study under him to learn better aspects of soil science, and I've gotten to do so much more that I never thought, soil scientists did or was possible. So it's been a really cool time. And then I recently had a baby, and so I've learned that it really takes a village. So the family helping out has really been helpful. Funding sources acknowledgement: without the professorship endowment from Dr. Wild, this project would not have been possible. And I'm very thankful for the additional funding I've received. And then with that, I will take any questions.
I'll ask a question. Spatial soil manganese maps, maps that you showed.
Yeah.
Were those data for those maps, at what sort of period of time were those data collected across the nation?
They vary. So I would say the most recent, that they get updated every year. There becomes a release of the soils data that's updated. However, the soil series data, that they do different projects in different regions, and then that gets updated maybe every 10 to 20 years or so, that they may look at that. Some regions, the last published soil survey was in the 1970s, and that may be the latest soil survey available, but most of it is more recent than that. The 2000s or greater, I would say, but I don't know for sure the answer to your question. I'm just telling you my best judgment answer.
Okay, if I may follow up, that is basically, I was just thinking, you were showing how the moisture and all that affects, manganese being sort of being designated or being redox conditions being created. Whether these variations in climatic conditions, moisture conditions from one year to another, sort of could compound the results that to be comparative on the same page.
I guess I would say that from what there was a pre-pilot experiment we did where we didn't get any manganese features to form, just using natural soils, and we concluded that it probably takes a very long time for these features to form. And I would say that probably very small seasonal variations could influence the overall picture, but that may be, 10,000 years from now. So you need to have, an average condition of that to create the features, and the features you see in soils are probably quite old and have been subject to pedogenesis for quite a while.
I was wondering on your maps, in your first study, it looked like Illinois was a lot of the turquoise, and all the other states around it were the dark blue. And it looked like they almost followed state boundaries. And I was just wondering, that was a pattern that was again and again, and I would think would have similar parent material, similar climates, similar topography, similar drainage. So I was just wondering what is there that cause so much more manganese to be in Illinois? I mean, it is a Democratic state, the others are Republican around it. Maybe there was a moratorium on manganese, I don't know. There's I just wondered if there's an explanation why it's different than what, you know, you see it kind of sticks out like a sore thumb.
A little bit, but there's also these other areas here.
Well, down in
So-
Arkansas, yeah.
Yeah, I hadn't noticed Illinois in particular sticking out. That could be one of those artifacts that I mentioned, and
I
that
mean,
could
it
be,
always follows the boundaries of the state.
yeah. Yeah, I'll have to look at that in a little bit more detail.
Yeah, just
Thanks for
curious.
pointing it out.
You know, I don't know much about what you're doing, but I know colors. You know.
Dr.
And-
Yar?
Oh,
Oh, sorry, I didn't mean to
no,
interrupt
no,
you.
I was just gonna, I noticed in your last study, you had a range of pH from 3.5, 3.9, all the way up to 5.9. I was wondering, did soluble aluminum have an impact on the oxidation reduction state or the KSP of the manganese compounds?
I don't know, but that's definitely something that I could look into
I mean,
and think
because
about.
You get
Yeah.
below pH 4, you have a real pickup in soluble hydrogen. And then from 4 up to
Yeah,
5, you know, there's, you know, when you drop from 5.5 to 5, your soluble aluminum goes up 60%.
right.
So you have a lot more aluminum and then even some hydrogen in those in the 3s. So I was just curious if that would affect your KSPs of your manganese compounds.
Yeah, it's definitely a thought. That,
Just
yeah,
curious, you know, I
Yeah.
mean.
it's definitely something to look into. Thank you for the thought.
Oh.
I'm gonna go to you because you,
Yeah.
and then over.
Yeah, so I was just curious about the interplay between carbon and manganese coatings and wondering, I mean, so you see these in the BTs, you see them when there's a carbon, and I'm wondering if you don't see these coatings because carbon is taking up those spots, like the organic material is taking up spots on the mineral surfaces, if there's some, I'm wondering if there might be thresholds there, what your thoughts are about that interplay.
Well, they occurred in soils that had low carbon. So, yeah, the presence of carbon for these features to form appears to be, needs to be low. And that there's some influence of the carbon that may inhibit the formation of manganese redox coatings. However, you know, with that experiment with the ISPs, you know, we only had 3 samples out of 6 different ones that resulted in that. So it needs to be tested for a wider range of soils.
So were those
However,
all
the-
subsurface soils?
Yes.
Yeah, okay, so
Yes.
you didn't do this, they weren't the surface horizons in any of them?
No, so I would say that, you know, the carbon content needs to be low for those features to form. And that maybe there might be some influence of the carbon content inhibiting those features, but the other soils that were tested, the Downer was well drained, sandy, very low clay content. Elkton didn't have a lot of manganese content already because it was poorly drained and fine textured. Codorus, I can't remember the manganese content on it, but it, you know, wasn't fine textured and it had a higher carbon content. So maybe that is providing some indication that the carbon is inhibiting the formation of manganese coatings there. Yeah.
These soft masses, I've seen this about iron, either pore linings or soft masses. And
Right.
Are they really soft? Like, what do they look like? Are they like little dark things or just like a sort of a low concentration accumulation in that area?
They're kind of like just splotches and they can be rather large, maybe like the palm of my hand, or small, maybe like, you know, like the size of your fingernail, and they can just be splotches, and they tend to occur more in floodplain soils with fresh alluvial deposits forming. So maybe they are characteristic of where, you know, these features are starting to form in younger deposits of here, but these younger deposits may still be quite old.
Do plants alter these things? That was another thought. Plants vary in that way too. Do they see things like the carbon input and or oxidation in the soil?
Right,
Is that something that relates at all to that?
with my literature review, I have found that there are some influences of carbon cycling and plant biocycling of manganese being uptaked in plants and then the leaves falling, and there being this sort of cycling process of manganese. However, that's more in the surface soils where that cycling is taking place and these features are forming in the subsurface. So the carbon cycling happening down there, I believe, is, you know, less of an effect because that carbon is being used at the surface with the microorganisms, and the carbon content in the subsurface is usually pretty low. Anyone else? Jason?
Accumulations, I was curious, is there like a time period where certain accumulations was used more?
That's a good question.
Why that judgment call is based on sense? That's not something that
Yeah, that's a good question.
it's focused on.
Yeah, I don't know, you know, they, with some of the terms that are used in these OSDs, they, you know, will say the, you know, the horizon, the texture, structure, you know, subangular blocky, iron concentrations found, manganese concentrations found along PED faces, 2%, you know, coarse in size. They kind of, you know, it's a verbose description, and it can vary based upon who's describing the soil of what was put in there. And so I'd have to look to see potentially like if the terms accumulations are in the OSDs where maybe they were updated a long time ago, because I do have the information of when the origin date of the soil series and then the last updated date, and that could provide some information on that. Yeah.
That's very interesting work. Thanks for bringing. So what are you going to do after you finish your PhD?
I'm gonna take some time off and hopefully publish some papers. So, yeah. And by the way, this is a friend from my previous graduate work at University of Delaware. She was in our lab, so it's like a friendly face in a new institution. It's cool. All right, I think we're done.